Hi, I'm Dr. Nathan Apson from the University of Sydney, and I'm going to be guiding you through this video abstract for our publication in Brain Communications titled Gain-of-Function Gabrb3 Variants Identified in Vigabatrin-hypersensitive Epileptic Encephalopathies. This is the story of 3 patients, all with genetic variants in exactly the same gene, but have very different reactions to the same drug, vigabatrin.
Case 1 is a 21-month-old girl who presented with infantile spasms at 7 months of age with hypsodinic EEG. She responded well to vigabatrin without any side effects. And in contrast to Case 1, Case 3 and Case 4 developed adverse effects on vigabatrin. Case 3 is a 4-year-old girl who presented with the spasms at the age of 5 months, and she regressed in development after the seizure onset. She remained refractory, and treatment with the vigabatrin caused extreme drowsiness and exacerbation of hypotonia, due to which it was stopped. Similarly, Case 4 presented with early onset infantile epileptic encephalopathy with the tonic seizures at the age of 3 months, with a background of severe global developmental delay. Introduction of the vigabatrin resulted in seizure cessation, but caused severe exacerbation of hypotonia, sedation, and respiratory difficulties. In both cases, the side effects reversed upon treatment cessation. Subsequent genetic testing in these cases identified Gabrb3 variants, and we asked our scientist colleagues if the adverse development of vigabatrin are a consequence of molecular phenotype of the individual Gabrb3 variants.
Gabrb3 encodes for a subunit of the GABA-A receptor, an inhibitory receptor that is activated by γ-aminobutyric acid to open a chloride ion channel. We identified 3 distinct Gabrb3 variants that were prescribed vigabatrin. Cases 1 and 2 had a truncation at position 194. This removes the transmembrane region to render the protein inactive. This is a loss of function variant, and Case 1 was seizure-free after vigabatrin treatment. Case 3 had a missense variant with a glutamic acid substituted for lysine at position 77 in the coupling loop. This loop is important in activating the protein. Case 4 was also hypersensitive to vigabatrin and had a threonine substituted for an isoleucine at position 287 in the transmembrane region that lines the channel pore. So how did the E77K and T287I variants alter receptor function that could lead to vigabatrin hypersensitivity? As the variants were de novo, we created concatenated receptor constructs with 1 copy of the variant or 2 copies of the variant to replicate the receptors that are created in vivo. We then injected the RNA into xenopus oocytes and waited 2 to 4 days for the receptors to be expressed. Here is the equipment we use in our lab to measure the activity of the wild-type and variant receptors using the 2-electrode voltage clamp technique. We used a semi-automated 3-chamber system for concentration response curve to GABA, measuring peak responses to GABA and fitting them to the Hill equation. We also measured the absolute expression, estimated open probability, and deactivation properties of the traces. All the synaptic gamma-2 beta-3 alpha-1 beta-3 alpha-1 constructs were robustly expressed, with only the E77K variant with 2 variant subunits displaying reduced absolute currents. Here are the results of the E77K variant. Regardless of whether the 1 or 2 variants were introduced, the concentration response curve is shifted to the left, and there is no change in the estimated maximum open probability. This means that it requires less GABA to activate the receptors, a gain-of-function variant, something we haven't seen before in Gabrb3 variants. The T287I variant displayed a similar phenotype, with a shift to the left in the concentration response curve and no change in the estimated maximum open probability. Furthermore, we found that E77K and T287I extrasynaptic gamma-2 beta-3 alpha-5 beta-3 alpha-5 receptors share the gain-of-function phenotype. So how could vigabatrin be causing hypersensitivity? Here I have a model of a neural Gabaergic synapse. GABA is constantly formed by the enzyme GAD and broken down by the GABA transaminase enzyme GATA1. When GABA is released at the synapse, it is quickly transported back into the presynaptic neuron by the GABA transporter GAT1. Vigabatrin disrupts the regulation of GABA concentrations at the presynaptic neuron by inhibiting GABA transaminase. GAT1 now reverses the transport of GABA, resulting in low concentrations of GABA persisting in the synaptic cleft, permanently activating synaptic GABA-A receptors. This is likely to be exacerbated in hypersensitive E77K and T287I receptors, leading to the clinical reaction to vigabatrin that was explained at the beginning of the video. We've shown that different molecular phenotypes will require different drug treatments targeted at the functional effect of the epilepsy-causing variant, paving the way for a precision medicine approach to the treatment of some forms of DEE. Funders that made this project possible.